All right, let's discuss the events at the neuromuscular junction. The neuromuscular junction. It's the space between the axonal end of a neuron and the surface of a muscle cell. Right there, that's called the neuromuscular junction. That's where all the magic happens, where excitation is going to take place. And I'm going to cover excitation events all the way to relaxation. So we're at the neuromuscular junction. And what I'd like to discuss first is the excitation event. All right, so think of a neuron has a cell body in the central nervous system and has a long axon and has, I'm kind of exaggerating the end of it. The axon terminal is this end, and it's on the surface, it arrives on the surface of a muscle cell. So this is the muscle cell. And this is what we call the neuromuscular junction. This is where things are going to happen. This is the sarcolemma. Which is a plasma membrane. It's the surface of the muscle cell. So this is the muscle cell, or myofiber. The myofiber. So we're discussing the events happening here. How does the myofiber get excited from the signal of a neuron? So I'm going to draw at the neuromuscular junction. I'm going to draw right here now in much large. What we have arriving at the end of this axon terminal, what we have is an action potential. An action potential is a depolarization of the membrane where we have positive charges on the inside of the cell membrane, negative charges on the outside. A regular potential at the membrane is normally negative on the inside and positive on the outside. All right, so this action potential is actually a depolarization at the membrane that is propagated all along the membrane, and it arrives right there at the end of that axon terminal. So when it arrives here at the end of the axon terminal, what happens is that it's going to excite excite voltage-sensitive channels. These are channels for specific ions that are sensitive to the voltage change at the membrane. If there's a voltage change at the membrane from the resting membrane potential to now a depolarized membrane, there's going to be a gate that opens up to allow the passage of ions across this channel. So in purple, we have those voltage-gated channels. So when the voltage-gated channels open, what's going to happen is that calcium is in higher concentration outside of the axon terminal. So as it sees this open door, it's going to naturally flow down its concentration gradient inside the cell where it's maintained at a lower concentration. So when these gates open, the calcium rushes down its concentration gradient. This is a passive transport into the cell. When the calcium enters the cell, calcium levels rise. When calcium levels rise, the vesicles that are inside the cell that are filled with a neurotransmitter come towards the membrane and fuse with the membrane and allow their content to spill out into the cleft. So this is the neurotransmitter in those vesicles, and that neurotransmitter comes out. And in the case of this story, it's called acetylcholine. So acetylcholine, I'm making it bigger and bigger, I'm sorry about that. So all these little dots are representing acetylcholine molecules, which are types of neurotransmitters here, and that's the neurotransmitter involved in the excitation of the muscle cell. So voltage-gated channels open, calcium ions rush down their concentration gradient, calcium levels rise inside the synaptic knob, vesicles containing the neurotransmitter acetylcholine come to the membrane, and by process of exocytosis release acetylcholine in that synaptic cleft. On the opposite end, we have the sarcolemma. So this is the sarcolemma of the myofiber. At this point, we still have regular resting membrane potential. So that would be negative on the inside, positive on the outside. That's a regular resting membrane potential, just like I wrote it here. Negative on the inside, positive on the outside. There's another video explaining the establishment of that membrane potential. How is it there to begin with? So this membrane potential is always there at the surface of the cell. If you tweak it, now the cell gets alerted that something needs to happen. So what happens is that on the other side of this surface, this area here, we have channels for different ions that are going to come into the picture now, but there are also channels that have a receptor site for acetylcholine. So I'm going to use green. These are ligand-gated channels. Ligand-gated channels, so in green, these channels open up only if a ligand binds on the receptor portion. Ligand-gated channels in green. So this gate, there's a gate here that would be closed across here, so I'm not going to draw it quite now, but not only is it a channel for a specific ion I'm going to mention in a second, it's also a receptor that's going to accept the acetylcholine as its ligand. So it's a receptor specifically for acetylcholine. So acetylcholine comes down here and binds on its receptor. So acetylcholine binds on the receptor. Acetylcholine is considered the ligand. It binds on the receptor for this ligand right here, and that's when the gate opens. So normally the gate would be closed, but the gate opens when the ligand binds on this receptor. So that's why we're calling this a ligand-gated channel. It's a channel that's always closed except when the specific ligand binds on the surface receptor portion. Okay, and what is this channel for? This channel is for sodium ions. Sodium ions, if you've watched the video about the membrane potential, sodium ions are always more concentrated on the outside than they are on the inside of a cell, because there's a sodium-potassium pump that makes sure to keep this disproportionate amount of ions across the membrane for the reason of establishing a membrane potential. If we keep these charged ions in uneven proportions across the membrane, we're able to have a surface that is slightly more positive versus a surface that is slightly more negative, and now you have a polarized membrane for the cell. And every cell is like that. It has a polarity on the surface that you can now change, and that's the purpose of it, is to be able to tweak that polarity. So here we have these ions that are coming into a compartment where they're less concentrated, and that's why they're flooding down their concentration gradient. They're flooding inside the cell. Now we're talking about the myofiber. So now here we are inside the myofiber. we have sodium ions that are coming in. So this is going to contribute to a local depolarization at the membrane. This is bringing in plus charges. So what used to be in the muscle cell, it's negative millivolts, right? If you took two electrodes, you poked one inside the cell and you left one outside of the cell, and you measured, it would give you a voltage in millivolts. That's the unit, and it would be negative 90 millivolts of difference between the inside and the outside of the cell, with the negative 90, the negative portion, referring to the inside of the cell. So the inside of the cell is negative, and it's there's a 90 millivolt difference between the inside and the outside of the cell. When you're adding plus charges, now the inside of the cell is less negative, right? Because you're adding plus charges, so you're neutralizing some of these negative charges. So now it might be negative 80, and then it's going to be negative 70, and then it's going to be, right? So the more plus charges you add, the less and less negative it's going to be. And at some point, it's going to reach negative 55. Negative 55 is the threshold. This is the threshold that is basically going to be enough depolarization for downstream players over here that are going to come into the picture now. So when the depolarization reaches negative 55, you have downstream channels that are voltage sensitive that are going to come into the picture. So I'm drawing them in purple because that's the color I used for the voltage sensitive channels that are up here. Well, we have also voltage sensitive channels down here in the myofiber, but they're just for different ions. So these voltage sensitive channels in purple here have a gate. The gate opens up, right? So this is a gate that opens up. The gate opens up to allow for the flooding of additional sodium ions. So that's what these channels are. These are voltage gated sodium ion channels, as opposed to the ones we saw up here. Those were voltage gated calcium channel ions. So here we have voltage gated sodium channel ions that are kind of hanging in the membrane, and they're really not doing anything. Originally, when the voltage was changing originally with the entrance of the sodium ions over here, they didn't open right away because they're wondering if it's still going to be worth it. They're waiting for enough depolarization to actually open up, and that's what we call the threshold. When the threshold is reached, that's when these voltage sensitive channels will open and allow for their specific ions to come in, and in this case it's sodium ions. So we have the entrance of additional sodium ions that are going to come in, which means we're adding even more positive charges. At that point, now we had enough positive charges that we're completely reverting the polarity at the membrane, and we have completely depolarized the sarcolemma, and we have recreated the action potential on the surface of this myofiber. And that's the part of excitation. We have the action potential on the neuron that has been reproduced on the surface of the sarcolemma. Now the muscle cell is excited, and the action potential is going to go forward on the surface. So in the next video, I will explain the next step, which is excitation contraction coupling.